I'm Reza Farivar. I'm the current director of the integrated program in neuroscience, and it is my pleasure to welcome you to the 2020 Bernice Grafstein Lecture in Neuroscience. This year's speaker is Professor Huda Zogby. And before we begin, we're going to have receive an introduction of Dr. Grafstein by Caitlin Eason. Following that, we'll have an introduction to Dr. Zogby by Professor Ilana Watt. And then following that, we'll have the lecture by Dr. Zogby herself. Professor Ed Ruthazer is also online. He's the Associate Director for the IPN. So without further ado, I'll pass the buck to Caitlin to introduce Professor Bernice Grafstein. Thank you very much.
Thank you, and good afternoon, everyone. My name is Caitlin Eason, and I am the president of McGill's Graduate Student Association for Neuroscience. Today's lecture is named in honor of the esteemed scientist who I have the pleasure of introducing, Dr. Bernice Grafstein. Just like many of us in attendance today, Dr. Grafstein was once a graduate student at McGill, where she completed a PhD in neurophysiology. Trained as an electrophysiologist, Dr. Grafstein's doctoral thesis work was centered on cortical spreading depression, a widely recognized phenomenon today that we know to play a role in migraine and stroke. Dr. Grafstein later developed a fascination with nervous system development and regeneration, interested in studying how the connections form between the cells of the nervous system. She trained in this field with renowned embryologist Victor Hamburger at Washington University before beginning her own research on nervous system regeneration at the Rockefeller University. Dr. Grafstein's work has been focused on the role of molecular signaling in nervous system regeneration, including the role of intracellular protein transport in normal and regenerating neurons. Today, Dr. Grafstein is a Vincent and Brooke Astor Distinguished Professor in Neuroscience at Weill Cornell Medicine. Beyond her work in the lab, Dr. Grafstein has been president of the Society for Neuroscience and is currently a vice president and trustee of the Grass Foundation, an organization whose mission it is to support and encourage education and training in neuroscience research. Thank you to Dr. Grafstein for your valuable contributions to the neuroscience community and for joining us here today at your alma mater for the 2020 Bernice Grafstein Lecture in Neuroscience. And with that, Dr. Grafstein will now say a few words.
Yes, when I was a graduate student at McGill, I could only dream of what my scientific life might eventually become. At that time, I didn't realize how important my McGill years were, and I certainly didn't realize how important they were going to be eventually. It was a time when, unbelievable as it may be, neuroscience as a discipline just didn't exist. There wasn't even such a word in the language. Now, McGill has had a remarkable record in neuroscience. It's produced many figures that have made classical contributions in the field. And I'm glad that by supporting this lecture, I can remind our students of the role that McGill has played and is continuing to play in this exciting field. And I welcome them to a wonderful future in science. Thank you.
Thank you very much. That was nice. I'm going to, now I have the great pleasure of introducing Dr. Huda Zogby, our keynote speaker. Dr. Zogby is a professor at Baylor College of Medicine and a Howard Hughes Medical Institute investigator. I want to start by talking about Dr. Zogby's career path and highlighting one early challenge that she faced that I think can serve as a model to inspire students and remind us of the importance of perseverance and resilience during these challenging pandemic times. Dr. Zogby completed her Bachelor of Science at American University of Beirut in Lebanon and started in 1976, and she started med school there later that year. However, civil war broke out, making it impossible for her to continue her studies. Dr. Zogby moved to the States for what she hoped would only be a short time, but when it became clear that she could not return, she persevered and she transferred to the Meharry Medical College in Nashville, Tennessee, where she completed her MD. She started at Baylor College as a pediatric resident and has been there since, becoming full professor in 1994 and the director of the Jan and Dan Duncan Neurological Research Institute in 2010. Over the course of her career, she has received numerous awards and honors. I can only highlight a small subset here, as otherwise I would take up too much time from her seminar. She has been elected to the National Academy of Medicine and the National Academy of Sciences and the American Academy of Arts and Sciences. She has received the Breakthrough Prize in Life Sciences, the Canada Gairdner International Prize, and most recently, the Brain Prize from the Lundbeck Foundation in 2020. Anyone working in the same field knows her reputation as an extremely supportive and generous PI and colleague. One example of this is the Zogby Scholar Award that she established, which is awarded to postdocs on the cusp of starting their career as independent PIs. Indeed, Dr. Zogby's proudest accomplishment is the successful mentorship of her trainees. She has over 90 graduates from her lab, including 5 in Canada, among whom is one of our newest colleagues, Dr. Wei Xiang Huang from the CRN. Remarkably, almost 3/4 of her trainees are in academia and 90% are doing research, which really is a testament to her skills as a mentor. So in addition to the work she'll tell us about today, Dr. Zogby has had an equally impactful career in neurodegenerative diseases, especially ataxias. She identified the genetic mutation in the ataxin-1 gene as the cause of spinocerebellar ataxia type 1. Dr. Zogby's lab actually made this discovery at the same time that it was discovered in Harry Orr's lab at the University of Minnesota. However, rather than competing, Dr. Zogby suggested that they collaborate, and they published their findings together, which was the start of a legendary collaboration that has endured for over 25 years, and which has been the source of inspiration for many colleagues in the field, myself among them. Today, Dr. Zogby will be talking to us about her groundbreaking contributions to the field of neurodevelopment. She's probably best known for the discovery that mutations in the methyl CpG binding protein 2, or MECP2, is the cause of the severe neurodevelopmental disorder Rett syndrome, a discovery that took 16 years to come to fruition. Talk about persistence. Her most recent work shows the promise of antisense oligonucleotide therapy as a treatment for Rett syndrome. I know that everyone here joins me in welcoming you to McGill, Huda. We're a little sad that you can't be here in Montreal in person, but we are happy that as a community, we can be resilient and persevere through these challenging times. And thus, we're thrilled to host you virtually for this seminar. Thank you.
Thank you so much. I really am honored to be here. I'm honored to give a lecture named after Dr. Graffstein. I so enjoyed our lunch together today with some of the students. And I will come back. I will definitely come back and visit Canada and McGill, I hope. I appreciate the very kind introduction. It's really an honor. I should just maybe make one little fact clear. I had reached out to Harry to collaborate before we cloned the gene. So it was actually unusual in those times. And we ended up discovering it on the same day, which was really very sweet. So it's been one of the highlights of my career, really working with Harry. Thank you. I wasn't sure what to speak about, so I consulted my hosts and I was told Rett would be good. I really thought about talking about neurodegeneration, but maybe when I come back to visit, we will do that. So today I will share with you some of the work we've done on Rett syndrome. But before I start, I'd just like to declare some commercial, I should say, relationships. I am collaborating with Ionis Pharmaceuticals, and they have funded us recently for a biomarker discovery for MECP2 disorders, and we collaborate with UCB Pharma on SCA1, and then I'm on these advisory boards. So I think many people know that my journey with research really was inspired by a single patient. And this is Ashley, who I met her as a pediatric neurology resident in my first year. And what struck me about her is her history. She was perfectly healthy the first 2 years of life, learned to do all the things little girls would do, even singing nursery rhymes and saying a few words and greeting her father when he came from work every day. But sometime after her second birthday, all of that stopped. And instead of talking, she became silent. Instead of using her hands to feed herself, to leaf through the book, she had these ringing hand movements, and she became withdrawn, and her balance became affected. And this happened over a span of a few years. So we saw her about 3 years after that and had unsteadiness, and eventually had breathing problems, hyperventilating and holding her breath. And I was really struck by that history. And it was at the same time that I met her that Bengt Hagberg published his first paper about Rett syndrome, describing the disorder in 35 European patients. And I want to show you a video just to see what these individuals look in motion, so that the students at least will recognize the pattern. This consistent hand-wringing movement is very unique really to Rett syndrome. There are not that many neurological diseases that I know about. And it was really, and here you can see them, they have these, what we call apraxia, which you have the motor function, you have the strength, but you can't move. It's sort of inability to perform a motor act at will, and of course, unsteady. So, and this disease affects all ethnic groups, all patients that we've seen from all over the world. And I think it was this hand-wringing movement that I would say, may have clinched it to me about Rett syndrome, because a week after I saw Ashley, I was seeing a child with cerebral palsy, and she walked in wringing her hands. And then as I reviewed her history, it was the same story, healthy childhood and then losing milestones. And that's when I went into the records of our clinic and found a few more patients and published the first paper on Rett syndrome, and really became fascinated with the disorders and was convinced it has to be a genetic disorder. Of course, there were challenges with this hypothesis, is that we are here in late 1988, where the genome wasn't mapped, nothing was really known about many genes on the X chromosome or any chromosome for this matter. And I was convinced this is genetic, but yet the disease is sporadic. It's usually one girl in a family. So you can imagine that most people were really skeptical about this being a genetic disease because it was always one in a family. What I think people ask me, what convinced me it was genetic, I would say it was the development of the disease. It was the cascade of things that children have went through. By that time, I've seen about 100 patients, and all of them had the same history. So I was really convinced only a genetic program could really lead to such a phenotype. And what's also most interesting about the disease, it's not degenerative. They start healthy, they lose milestones, they don't acquire new skills and stay in that state for decades. Some of the oldest patients are in their 60s and 70s. So because of the lack of genomic resources, if you will, and the technology back then, it really took time just simply because it was brute force sequencing gene after gene on the X chromosome till we finally identified the correct gene by Ruthie Emir. And it is methyl CpG binding protein 2. It's on the X chromosome, which explains why this disorder only affects females. And it encodes this protein that binds methylated cytosine discovered by Adrian Bird in 1991. And at the time, he identified it by purifying it through binding to cytosines that were methylated, especially when they're followed by guanine. So that's what was known. So what I'd like to share with you today is the following. I'd like to very briefly review with you the phenotypic spectrum of MECP2 disorders and what we learned from the various mutations, and this is really important. Then I will discuss some molecular studies recently on the function of MECP2. We'll move then to neuronal function of the protein, what happens when you don't have it to neurons, and conclude with some recent studies, unpublished work on the MECP2 regulation and our path to therapeutics for the MECP2 duplication syndrome. So what we've learned about this disorder is that majority of Rett syndrome cases are caused by mutation in this gene. I would say over 95%. And I suspect the remaining 5% are also probably mutations in this gene, but probably not a coding mutation, and perhaps that's why it's been elusive so far. And they typically go through the course that I shared with you about Ashley. And males are extremely rare, but a male with a totally inactivated mutation, null mutation, is typically very sick and they die within 1 to 2 years of life. However, what's interesting is that there are some milder mutations, and these are compatible with life in males. And in females, of course, they're very mild in females. They may show slight learning disability or maybe slight OCD or anxiety. But males with milder mutations can live into their 40s or 50s and typically present with mild learning disability, autism, anxiety, obsessive compulsive disorder, hyperactivity, even bipolar disorder, and juvenile schizophrenia. Now, these symptoms here or features are in different colors because these will be different in different patients. But the mild learning disability is common to all of them, and typically tremor is also common to all of them. So the point of this is the severity of the mutation really determines a phenotype. So null will cause severe phenotype and lethality. Mosaic, for total loss of function, if you will give classic Rett syndrome. And then very mild allele can give now more psychiatric spectrum phenotypes. So this is something, it's much easier to learn from human patients because you can imagine it's really hard to have mouse models for juvenile schizophrenia. But the mouse model does replicate all the typical neurological features of Rett syndrome. And you'll see on this video a healthy mouse, and then you'll see the mouse with Rett syndrome with the typical forepaw movements. It's reminiscent of the hand wringing, and the mice have all the other features that we see in the disease. So when we first identified the gene, one thing we wanted to know, would adding the gene rescue the disease? As a positive control for gene therapy, we decided to put a large insert clone using a modified pack, which contains all the coding exons of the genes and the regulatory, what we thought were the regulatory elements, because we basically used a large clone that contains only the MECP2 genes and none of the neighboring genes. And this ended up actually reproducing the normal expression of the protein and the RNA. And these mice had their own endogenous copy, but they also had this extra human copy. And we had a couple of alleles, one that expresses twice the normal level and one that expresses 3 times the normal level. And to our surprise, these mice had severe neurological problems. They also had behavioral problems, anxiety and social behavior-like deficit, cognitive deficits, stereotype behavior, epilepsy, and even premature death. So that was really our first clue that doubling the level of this protein can also cause neurological disease. At the time, we didn't know of humans that had neurological disorder and duplication. And this made us revisit patients in our DNA lab who presented with phenotypes similar to Rett syndrome, but not Rett. And we started finding the duplication. And also Dr. Vanesh has actually reported on very nice series of patients, and all of her patients had typically these features. And it was a very nice case where the mouse actually predicted a human disease, and it's very true to the features of the human disease. We know that the patients have very complex rearrangements, so it's not always the same beginning and end for these duplications. And what you see here in red is the various patients with duplications, and then in blue, some with triplications. And you can see some of them are complex, so there's a region duplication, region triplicated. And this is work from the Lubsky lab characterizing this rearrangement. But what you'll notice is the one region common to all these individuals is this gene, the MACP2 gene, and then is another gene, a kinase, IRAC1, that's right next to it. And in our mice that reproduce all the features of the disease, IRAC1 is not included. Therefore, we do believe that most of the phenotypes are due to the duplication of the MACP2 gene itself. And by the way, none of these, the size doesn't quite correlate with the phenotype. We believe it's probably more the level of the protein that does. So then what we've learned from all these studies from human patients and then mouse studies is that this is a protein that's, the brain is very sensitive to its level. So you have to have it just right. And if it's absent, it's fatal. If it's in 50%, missing in 50% of the cell causes Rett syndrome. We have a mouse model that makes 50% of the protein, and they're similar to the Rett mice in males. And then I mentioned to you these mild mutation. This mutation has been seen in multiple cases of juvenile onset schizophrenia and bipolar. And then on the other side, duplications are severe. And I didn't tell you about the triplication mice, but they're more severe than the duplication. And the triplication humans typically die within infancy, whereas the duplication individuals would live into their second or third decade. And once we've known that both the loss and the gain causes disease, we began to investigate the mechanism and quickly learned they really have opposite effect on neuronal function. In this particular slide, you see number of synapses where loss of MACP2 causes decreased synapses based on this glutamatergic synapse labeling compared to wild type, whereas the gain of the protein causes increase in the number of synapses. And as I mentioned to you, the protein is known to be methylcytosine binding protein, and Adrian Bird and others have identified some of the complexes that associate with the protein. And one of them is the Encore TBL1 complex. And we have really thought it's going to be a straightforward story that this would be a repressor. But when we studied the gene expression in both the loss of function and gain of function mice, first, we see the inverse, just like I've shown you, the synapse phenotype is opposite. The same is true for gene expression. Majority, 95% plus of the genes altered in these models are oppositely misregulated. And in the null mice, we see a lot of genes that actually go down, and those same genes will go up in the duplication. So I believe the effect of the loss of this protein in gene expression is quite complex, but we can safely say that the duplication causes disease by a gain of function phenotype, and that gain of function involves the intrinsic function of the protein. Now, more recently, our understanding of DNA methylation has been expanded. And as you know, the DNA methylation is written by an enzyme, DNA methyltransferase, and typically the mark is read by a reader, with methyl CpG binding protein being a binder of this mark is one of the readers. And then, of course, there's the enzyme that remove the mark. These are the erasers. So about 7 years ago, Margaret Burns and Joe Ecker identified another type of DNA methylation that involves non-CG. So the one that has been studied for decades is the CpG methylation. But non-CG methylation, that's when the nucleotide following cytosine could be any of these. And what they found is that this mark continues to increase both in mice and humans as the brain mature. And we became interested in this because, as I mentioned to you, Rett girls are healthy for quite a while, typically 1 to 2 years before they develop symptoms. So we were wondering if this might explain it, because the mark is not present extensively at birth. It continues to be increased afterwards. Maybe this is why we don't see a phenotype in Rett till later. So we began studying the effect of this mark and whether this mark contributes to Rett syndrome. Now, we knew from multiple studies that DNA methyltransferase 3A is the writer of this mark in the brain. What we did not know is how much this mark contributes to Rett and whether MECP2 is the sole reader for interpreting this mark. So to do this, Laura Lavery, a postdoctoral fellow in the lab, began a very systematic study comparing the phenotypes of the writer, DNMT3A knockout, to the reader, MECP2 knockout. And she did the analysis specifically in inhibitory neurons and assessed everything with great depth at the behavioral, physiology, and molecular level. And what you'll see from this, I'm just going to show you a couple of data slides, is that the DNMT3A knockout mice, both males and females here, die much early. Their body weights, first of all, they die early, of course, but their body weights are much more severely affected than the MECP2 knockout mice. So from the outset, we see that those are more severe. Now, DNMT3A is an autosome, whereas MECP2 is an X chromosome, and here we're comparing males, so that's why you only see the data for the males. If I was to summarize the data, you'll notice that they share quite a bit of their phenotype, a lot of the motor phenotypes and coordination, learning, premature lethality, but there are some distinct, where we saw more severe weight loss, earlier lethality, and sensory processing defects in the DNMT knockout mouse. So with that, we decided to look molecularly and ask the question if MECP2 is the sole reader of this mark. So basically, when you lose DNMT3A, you still retain most of the methylcytosine CG methylation, and MECP2 can still bind these sites. Whereas when you lose MECP2, DNMT3A will, of course, methylate non-CG, and you have the other cytosines, but now we lost the reader. And the question is, how much overlap? If they are one-to-one, if MECP2 reads all the MCH mark and DNMT3A writes those marks, there should be a lot of overlap in the gene expression changes. So when we did this comparison and she purified the striatal inhibitory neuron nuclei, so this was a very, head-to-head comparison, what you'll notice, there were only 86 genes that had the same expression changes in both knockout. And the directionality, if the genes, half of them went up and half were decreased, was the same in both of these units. So we believe those two are really shared with that. But what's interesting is that these genes make up about 40% of the significantly altered genes in those neurons in the MECP2 knockout. So this told us that this methyl mark is really important for Rett pathogenesis. On the other hand, these genes only made up about 12% of all the genes altered in DNMT3A. So this finding raises the possibility that perhaps there are other readers for this mark. So today what we think is that these two do have a functional relationship, and they share many phenotypes when both are deleted from inhibitory neuron. But we think that MECP2 may be a partial reader of the DNMT3A methylation, and we believe the MCH, and it's predominantly MCA, significantly contribute to Rett syndrome pathogenesis. Now, moving from the molecular studies to some neuronal studies, over the years, we've studied the role of this protein in various neurons. And in summary, no matter which neuron we delete it from, the neuron becomes dysfunctional. It basically functions, the best way to describe it, it functions at about maybe 70% of its normal capacity. And I say that because we measure, we do the physiology study, and we see that the responses are decreased by about 30%. But just to make this point here, I'd like to share with you some interesting features we learned that may be relevant to people studying autism or other psychiatric symptoms. I'm just going to summarize for you what we learned from deleting this gene, either from glutamatergic neurons or from inhibitory GABAergic neurons. And this is work done by Shanling Meng for the excitatory neurons, and by Tuan when she was a student in the lab, and Aya and Kirsten when they were postdocs in the lab. And Kirsten is actually now in Canada. She's one of the trainees who's in Canada. So what they've done in this study, not only a deletion, but also they've done a rescue. So you bring the gene back only in those neurons. And this is what we learned from these studies. And again, I'm just gonna summarize here. What we've learned is that there are actually some shared phenotypes, but also some distinct phenotypes. For example, obesity, motor coordination, and premature death in males was common to both knockout. So this tells us they're both inhibitory and excitatory neurons that really are involved in these phenotypes. But what's interesting is that repetitive behaviors, learning and memory deficits, social deficits were apparent in the inhibitory knockout, but we did not see them with the excitatory knockout. In contrast, we saw the anxiety-like behavior and the tremor in the excitatory knockout. Now, altogether, these are all the features we see in the constitutive knockout of the gene. So these two neuron types clearly recapitulate all the features, but this partial disablement of the excitatory neurons, the first symptoms you might see are the anxiety-like behavior and tremor, whereas here you might see the social behavior and cognitive behavior. And then what was really interesting is when we looked at parvalbumin and somatostatin neuronal knockout, now we saw subsets of these phenotypes. So what this is actually telling us is that when a neuron is partially disabled, you will initially see the features of the symptoms that are the most robust sign of its vulnerability. And then, of course, as the disease progresses, you might see additional symptoms. And imagine now when you delete this gene from either inhibitory or excitatory neuron, one is getting all these features and death. Imagine what will happen if you now decrease inhibitory signaling and excitatory signaling by 10 to 20% rather than 30% that we see upon deletion of this gene. This might give you an idea of some of the psychiatric or milder features one might see. So having established that this is really important for every gene, for every cell type, and having established that so many genes were altered in this disorder, that's when we started thinking about alternative approaches to maybe think about how we're going to handle this disease. And one of the things we contemplated was using deep brain stimulation, manipulating the circuit in a specified region with focus on a particular phenotype. And for this, we collaborate with Zhenrong Tang, who's the director of our in vivo physiology core here, and at the time his postdoc Shuang Hao was wonderful, and she did all the deep brain stimulation in these studies where she stimulated the fornix, and we know, of course, that the fornix has projections to the hippocampus, and was able to record and find that she is getting the actual response in the dentate gyrus without any seizures or any abnormal responses. So what we wanted to see with stimulating the fornix will help the hippocampal phenotypes in Rett syndrome. And the paradigm she used is to wait till the animals are symptomatic, and they're typically female Rett mice are symptomatic around 2 to 3 months of age, and then to do the chronic deep brain stimulation following the same protocol that are usually done in humans, and communicated with the neurosurgeons, and then assess all the phenotypes that we knew are altered in the Rett female mice. So they had spatial learning deficit and contextual fear learning deficit, and they had abnormal LTP and decreased neurogenesis. And here's just, you'll see one example of the contextual fear learning, where the animals is typically put in a cage, it gets a foot shock, and the animal will recognize the space as which it got the shock. When it's brought back to the same context, it will freeze, which is a sign that it learned this is where the animal received the foot shock. However, if it's brought back where it hears the sound, that's a cue dependent learning that's more amygdala mediated. And what you'll see here, we know that in rat mice, the contextual learning is altered. You'll see that the rat sham mice have decreased freezing compared to the wild type sham mice. And that deep brain stimulation slightly improves the wild type mouse performance, but clearly improves the Rett syndrome mice performance. So with that, we then went on to measure in vivo long-term potentiation, and you'll see that the Rett mice over several days have decreased long-term potentiation, indicative of the decreased plasticity in the Rett brain compared to wild type. But after deep brain stimulation, this corrects, and here you see them pretty much up to the level of the wild type. And we've identified also improved neurogenesis at an additional phenotype. There's remarkable decrease in neurogenesis, which there's increase in both actually, the wild type and the mutant, but the mutant now have corrected to the wild type level. And these neurons could get incorporated in the network. We are now following up on this to see if inducing neurogenesis by itself is sufficient to improve learning and memory, but we don't have those the answers yet. Lastly, we looked at gene expression after deep brain stimulation, and what you see here, this is work by Amy Pohodish when she was in the lab. You'll notice that the Rett mice have quite a few genes that were different than the wild type animal, but after deep brain stimulation, they become very similar to the wild type stimulated animal. So we did note that many neuronal and synaptic genes were corrected. I'd like to mention here, just as a side, we looked at many of the genes that improved after deep brain stimulation, and we noticed that quite a few of them are altered in other intellectual disability models and in major depression in humans. So this raises the possibility that stimulation of the fornix might actually help more than one disorder with intellectual disability. And actually, Jean Rong has gone on and studied that in CDKL5 and showed it helped the learning there as well. Now, more recently, we wanted to see, would stimulation in other parts of the brain be helpful to additional Rett phenotype? And in this case, the stimulation was in the dorsal striatum. And this was chronic DBS, against the same paradigms as used in the clinic. And what you'll notice here is there's some improvement after this paradigm of deep brain stimulation, where you see the Rett animals fall typically quickly off the rotor rod, and their performance is intermediate between wild type and those that don't undergo stimulation. And here, additional test where you measure their motor function going down a pole test. You'll see that they do poorly. It takes them longer to descend, but again, after dorsal striatal stimulation, they improve. And this is walking on a thin dowel. They fall quickly, but that also improves. So I think altogether, what we think is that at least stimulation of the fornix improve hippocampal phenotypes of the dorsal medial striatum, improves motor function. So we can say that the Rett brain, at least in mice, is responsive to neuromodulation. And we're thinking now of ways how to test if the human brains would be responsive. It's not an easy task because, you know, these are acute studies to know if there is a response. But when you're looking at learning and memory, you can imagine a Rett child who's been unable to learn and in their state for many years. It's going to take a lot of training. So this is why we moved from cognition to motor, thinking if we see improvement in motor with deep brain stimulation, maybe this should be our first foray into a human trial so that we can easily measure motor improvement, more easily at least than cognitive that would require a lot of training. So I want to come back now to this slide where I showed you how important McP2 level and share with you a new story that was inspired by this. Recognizing that MACP2 levels matter, we realized that it must be very tightly regulated, and we thought it would be important to learn how is it regulated at the transcriptional level. So to do this, we collaborated with Josh Wyeth's lab here at Baylor, who was doing ATAC-seq in both developing brains and adult brains, and Yin-Yao Chow, who is a graduate student in the lab, analyzed the data, and particularly for MACP2, and identified various peaks that somewhere in the introns, somewhere upstream of exon 1, that were indicative of potential regulatory elements. But of course, to know for sure that any of those predicted based on ATAC sequencing, she had to now study them functionally. And for that, she generated various knockouts for these elements. Peak 4 is the promoter, so we knew this was established as it was already known to be the promoter for this gene, so we did not delete this. We knew it will create a null allele, but she deleted all the others and generated mice for that. And what you'll see here is the amount of MACP2 mRNA in mice in normal versus knockout of each of these peaks. And as you'll see, some of these peaks clearly reduce the RNA, while one of them increased the RNA of the gene. And then when we follow that with the protein, again, you'll see a corresponding decrease in the protein, and peak 2 had the most reduction for the protein level, whereas loss of peak 6 resulted in an increase of MACP2. So with that, she then went on to see, are these conserved in human? And she created deletions in human iPSC-derived neurons. And here again, you'll see that deletion of peak 2 results in about 50% reduction of MACP2, whereas knockout of peak 6 resulted in an increase in the RNA as well as the protein. So you see here the peak 2 knockout causes also reduced protein, whereas P6 causes increased protein. So now we have these two peaks that affect the protein level. And since I mentioned to you that protein levels must matter, you would predict that these animals should have some phenotypes that are similar to the knockout and the reduction, and similar to the duplication with the increase. And here's a couple of examples where you see in this anxiety assay, the animals who are anxious will go less into the open arms, and these animals are anxious. You'll see them spending less time in the open arm. And this is when we normalize to the distance traveled. And with social behavior, animals that have autism-like features or decreased social behavior will spend less time with the mouse, and it's not significantly different from an object or the cup. And you'll see here, wild-type animals are quite different from how much time they spend with the cup, but a little bit less though for these animal deletions. On the other side, the animal with the peak 6 knockout, this causes increase in the protein. They have decreased activity, and they have increased contextual fear, which is what we typically see in the duplication mouse. They have hyper response to contextual fear testing, and they freeze more. So I can't show you all the data, but I'll summarize them here. So this big circle is all the features of the Rett mice, or loss of function. If you have reduction of MACP2, you see all these features as we see in Rett syndrome. And what you'll see here with this peak 2 knockout is a partial phenotype. So of all these features, it's the hyperactivity, the anxiety, the social deficit. And this is reminiscent, if you recall when we discussed the human patients, this is reminiscent of the milder mutation causing more autism, ADHD, anxiety. So imagine this being mimicking a milder mutation. On the other end of the spectrum, we knew that the duplication causes all these phenotypes, but what we see here is this peak that causes just 50% more of the protein, causes a subset of all these features, which is the hyperactivity, anxiety, and learning and memory deficit. So there are a couple of things I'd like to highlight here. One is that the spectrum continues. I imagine if you add a little bit more, you're going to pick up more features here, and the same is here. Second, those regions that we identify now as enhancers for this gene are likely to be hotspots perhaps for mutations in autism or some of these disorders with some of these features. So, and those typically are not sequenced because they're in introns. So I think this would be really helpful to look at these regions. So now we've expanded the curve to really add the 70% and the 150% about all the possibility. And to conclude this talk, I'd like now to share with you our studies on moving into therapy for the duplication disorder. And this is work started by Hezi when he was a postdoc in the lab, and he's currently at Regeneron, and completed most recently by Yin Yao. So if you recall again, the mouse with the duplication had one human gene and one mouse gene and all the phenotypes of the duplication. And when we used an antisense oligonucleotide, this was developed by Ionis, we were able to correct quite a few of the features of these mice. So you see here the mice were hypoactive, and with ASO, that hypoactivity corrected. The same for their rearing episodes, their entries to the center. Many of the behaviors we studied were corrected. And I just want to highlight something important in this slide. We wanted to see if we treated them quite late, at about 7 to 8 months, at which time they're having seizures every day, what would happen. So we took older mice and we gave them the antisense oligos in the ventricles for 4 weeks, and we asked what would happen. And you can see here they have seizures all the time. Both we can see those clinically by videos and by EEG, and all of that corrected. So this was really promising to us, because doing studies on 2-month-old mice, one would worry, would this translate to an older patient? Doing it on a 7-month-old mice, we still worry whether it'll translate to an older patient, but at least it gives us promise that hopefully, even if we treat, because at this time they're having seizures all the time, it'll give us hope that maybe at this time we can at minimum reduce the seizures for the patients. So this was completed and gave us hope that this is a path for therapy. The problem is, as I mentioned to you, these mice had a mouse copy. So no matter how much antisense we gave them, we only deleted the human gene and the mouse gene was protective. But humans have 2 copies that are identical. So we had to make a new mouse model for us to have a path forward and provide a proof of concept for a trial. And in this case, created now mice that have 2 copies that are similar. We had GFP on one of them to differentiate them. And this allowed us now to see, can we titrate the dose of the ASO? Because you don't want to over-treat and cause Rett syndrome. If you give too much of the antisense oligo, you will cause Rett syndrome. And what you'll see here is that this is the mouse with increased level of MACP2, and based on the dose of the ASO, you can nicely titrate to reach the desired level. So with that, we then studied the pharmacodynamics of this treatment, and we learned some interesting things. What you'll see here is when you give the antisense oligo, the RNA within 1 week goes down, and it's still down at 2 weeks, and it's gradually is going to go up as the ASO wanes. The protein lags behind. The protein has a longer half-life, so it takes a little bit longer to come down. And when we look at genes that are typically misregulated, we see that those genes begin to change quite early on. So here's a gene that's normally upregulated, and you can see its level decreasing and lasting a long time. We then studied the behavior of these animals. These animals are opposite of the Rett mice. The Rett mice fall off. These stay longer on the rotarod, and you'll see that's corrected upon treatment with the ASO. I mentioned to you these animals have more freezing on the contextual fear, and with the higher dose, you'll see that behavior corrected. Now, one behavior that did not correct is the anxiety-like behavior. You see these animals have less entries into the open arms, and neither the low nor the high dose affected that. So I believe that either this would require more treatment, or that when an animal experiences anxiety early on in life, this may be harder to reverse. We don't know that yet. More work has to happen in this area, but at least it shows that some phenotypes might respond, like the seizures or the motor. Some phenotype, like the anxiety, may require additional therapies, behavioral possibly, or might take longer time to correct. If I was to summarize for you all the data we learned from these new studies, there are a couple of things that are really important. The mRNA normalizes quite early, as I mentioned, the protein a little bit later. You'll see the protein here. And some of the targets also happen relatively early, but the behavior does not correct early. What we notice that even though the protein is normalized at 2 weeks after treatment, the behavior does not normalize till about 9 weeks. So this is quite interesting, which we are using these studies now to really learn something about what happens between normalizing the protein and behavior or neurological functional change. So we believe there must be a cascade of events, and we're now focusing on those events to study them. From a practical treatment side, this is good, because this tells us in case we overcorrect, we have time before we cause neurological problems. If we can find a biomarker that we can detect early on, we have time to immediately reverse the antisense therapy using an antisense to the antisense. So we're now building on this, and to really translate the ASO therapy into clinical trial, we've done all the necessary steps. What we need now is a biomarker that at a single patient level, you can measure and say, this is it. I hit the right dose, we're safe, or no, we have to titrate the dose more or less. So we're at this step right now, and we're doing that in preclinical model, but our colleagues in the clinic who are involved in a clinical trial readiness studies with Ionis are doing that in human patient. And the biomarker does not have to be a molecular biomarker, it could be also physiological. So with that, I'd like to just acknowledge the wonderful people in the lab who's contributed to this work. Ruthie, I always thank her, even though she's long gone from the lab, but she really stuck with me and believed in pursuing the genetic basis of Rett syndrome. So she was the one who identified the mutation. I mentioned all the others as I went on, and Laura Lavery, the DNMT3A story. And special thanks to our collaborator, Jen Rong for the DBS, and Josh for the enhancer study, our computational biology colleagues and Ionis colleagues. And of course, my greatest gratitude is really to the families. They've been very patient and stuck with us all these years to learn, to help us discover the gene by participating in studies, and now are willing to participate in these biomarker studies. So it's been really a privilege to work with, and of course our funders, I should mention, HHMI, the NIH, and the Rett Syndrome Foundations, RSRT, and the families through the 401 project. And with that, I'll be happy to take questions.
Thank you very much, Professor Zogby, for that incredibly illuminating and interesting work. Now taking questions, it might be easier if you raise your hands on the on Zoom. Use the hand raising participation. The first question is from Alice. Go ahead.
Hi, can you hear me okay?
Yes.
Perfect. My question is more in regards to the last part of your presentation about the antisense oligonucleotides. Is the hope for translational studies to administer those intranasally for the patients, or what is the view that you have?
Right. As of now, given the current practices, it's administered via the the ventricle or intrathecally. It could be via a spinal tap or via the ventricle. And I imagine it would have to be done at least twice a year, given the current state of antisense oligos. So it is invasive until a better method of delivery comes through. That's how it's being done for the spinal muscular atrophy.
But have you tried the studies doing it with mice, doing it intranasally?
I have not, because our ASOs come from Ionis, and at this point, they only work if you put them directly in the CNS.
Okay, thank you.
Thank you very much. There's a question in the chat from Javid. I don't understand how DBS remedied the symptoms in the red mice and how it had effect on the DNA sequence of the neurons. Also, I can understand how ASO could decrease the gene expression in mice that have too much expression, but not how ASO treatment could increase gene expression in deficient mice.
Both are good questions. So how the DBS corrected the gene expression in the rat mice? It's interesting. As I mentioned, is the rat neurons, for one reason or another, everything we measure about them, when we measure their neurotransmitters or we measure their excitatory or inhibitory currents, they're always functioning at a reduced level. It could be that this extra stimulation has helped boost their activity, and that in turn improves some of the gene expression changes. That's really the best scenario I can think of why DBS have corrected about a good number of neuronal genes in these mice. Regarding ASOs, ASOs are only for the duplication. We are not using these for Rett syndromes. The ASOs are used, as you said, to lower the protein. For Rett, right now, we don't have an equivalent DNA-based treatment. The best you can think about is maybe gene therapy, but with gene therapy, as you know right now, for non-human primates or any animal larger than a mouse, you're only hitting 10 to 15% of the neurons. And you also have to be mindful with gene therapy that you have to give just the right dose, because if you give too much, you have wild type cells. Half of the brain is wild type cells. So the studies are still being fine-tuned for gene therapy. With ASOs, while you worry about overcorrecting and going too low, but you can always give an antidote. You can give an antisense to the antisense. Whereas with gene therapy, once you give it, it's pretty tricky. You're going to have to chronically treat to lower it again. So that's why that has to be fine-tuned.
Thank you again. I invite everyone to either post your question in the chat or raise your hand. So Alana?
Hi, can you hear me?
Yes.
That was a great talk. Thanks so much. So I have a question about ASOs that I've actually long wondered about. Do you have good evidence that they're getting to all the cells equally well, or do you think that there's difference in penetration with that? Also, a follow-up question actually about the difference between the behavior and the changes in the protein level. Have you looked to see how, like whether the circuits are being sculpted? Like, have you looked at all those other things that you've characterized along the way? Because I could imagine that circuit changes take longer time.
Yes, absolutely. Thanks. Great questions. This is the problem. When I'm trying to tell too many things, I don't show in depth something. So let me share with you. In the mice, when we use these ASOs, we got it beautifully to distribute throughout the brain. We went brain region by brain region, doing not only IF for the ASO, because you can use an antibody, but also we did quantitative analysis of MACP2 levels, both at the RNA, which is more precise even. So we see it distributed beautifully everywhere. So of course, the first thing we looked at is the amygdala. I want to know, why didn't we rescue the anxiety? Well, MACP2 level was perfectly normal in the amygdala after ASO treatment. So that was not our problem. And thank you for asking that question. So the second question is, what happens to plasticity? What happens to circuit? And all of that normalize. It's really beautiful. So these mice had increased LTP, the opposite of the Rett model, and the LTP normalized beautifully. So we do believe, and as I mentioned to you, even the EEG normalize at an older age. So pretty much everything we saw normalized. The only thing that intrigued us, and this is what we're now focused on in the next phase of research, is we normalized the protein, but it took 6 to 8 weeks for any functional change to become apparent. While the protein is back to normal and some of the gene expression changes are getting back to normal, it does take time for the recovery. So to your point, why is it taking time to recover? Is it taking time because you need almost all the genes to be normalized to see a neuron functioning robustly? Or is it because you actually normalized everything, but the neurons have to take time to be resculpted, to look at their morphology, their synapse number, dendrites, physiology. So we're doing a timed experiment now to assess all of that week by week.
Thank you.
Thank you very much. There are more questions in the chat. So was DBS tried in humans?
DBS is tried in humans, of course, for a variety of disorders, particularly Parkinson's disease and tremor. But it's this particular paradigm, I should have mentioned this, is being tried for Alzheimer's disease. Stimulating the cortex to improve hippocampal function is in clinical trials for Alzheimer's. Of course, in Alzheimer's, the neurons are gone, the cholinergic neurons and the fornix are gone. So I don't know how much benefit will be. That's why we thought in Rett it might work better because the neurons are still there. But it is being tested in humans, in Alzheimer's, but not yet in Rett patients. And again, I mentioned why, because I really, it's hard for me to imagine how you can do something that you have to measure over 2 to 3 months and expect to see such great cognitive improvement. You're going to have to train and do so much more. This is why we pivoted to do motor, because we thought motor you can at least assess a little bit easier. It still might require a lot of training.
Thank you very much. Another question is, do you have some idea on if and how histone modifications are affected by ASO?
Good question. We don't actually know how or if. We don't even know exactly how and if and what histone modifications are altered in Rett syndrome, even when you lose the protein at a very precise level. So back to the experiment I mentioned to Alana, we're adding those studies. We have a paper that's coming out soon. I didn't have time to present that shows the chromatin structure and some of the histone mark, like H3K27 trimethyl, are increased in the Rett animals and decreased in the MAPP2 duplication animals. We find everything always the opposite. You have more condensation of the chromatin structure and the DAPI foci, heterochromatic foci in Rett. You have it more, if you will, looser or amorphous in the duplication. So that paper will be in Journal of Neuroscience. But binding and what's happening, we're doing that now.
And another question is, are the behavioral phenotypes in the peak6KO animals pertinent to a particular sex of the animal? The iPSC neurons, are they region-specific, striatal, or hippocampal?
Right. So the answer to the first one, we see that what we did, all of that is in males, because we knew that a 20% change or a 50% change is not going to be very prominent in a female mosaic brain. So we looked at that only in the males. So this is why I think if I was to bet, is maybe MAPP2 is one of the risk factors why we have more males with autism than females, perhaps, right? It could be an X-linked factor. It could be MAPP2, it could be another X-linked gene. So we looked at males. The iPSC neurons, these were from humans, fibroblasts that were then converted to iPSC neurons and then just made glutamatergic neurons. They were not region specific.
Thank you again. I invite others to ask questions either in the chat or by raising your hand. Please don't be shy. This is a rare opportunity.
And the questions are so wonderful. I mean, obviously what you're asking are the aims of my NIH R01. So,
well, we do have the best neuroscience students in all of North America, if not the world. So, yeah.
I believe that. And I think that if any one of them is interested in following on these questions, we have the projects going, just started.
For postdocs.
That's right.
Other questions?
We'll take them as postdoc and send them back.
Perfect. That's exactly what we're hoping for. Did you try the effects of DBS in the ventral striatum? That's another question that's been posed.
No, we have not. We have not. It's actually tricky. It was really tricky what to try in the mice. Unlike humans, where certain targeted areas, like the subthalamic nucleus, would have been nice to try because they have a lot of movement disorders, but it's almost impossible to do in the mouse. So there are some challenges. So it was really, it took a lot of thinking, and we only tried the dorsal and we got really nice results. It's obviously endless what one can try, but we wanted to see if we can see something else besides the hippocampus. So that was encouraging.
I have a question as well, actually. Has there been studies on just if basically what the net effect of the anxiety is on just therapy or any kind of therapy for Rett syndrome? So basically, if the patients are exhibiting extreme anxiety or strong anxiety, how much of their inability to learn or to engage in therapeutic
Right.
prevents that?
Great question. The question is, could the anxiety be contributing to compromising some of their abilities? And it's really an important question. It's always bothered me, you know, to see them anxious and to see them, because sometimes families will tell you when you take them out of the house, they will get really anxious. If you take them to the mall, they start screaming. So obviously they do get anxious very quickly and very easily. And when they sleep a lot, all the abnormal movements and the breathing pattern that's altered, all of that disappears. So during sleep, their hand wringing stops, all of that. So obviously their awake state and whether the anxiety contributes to that is somehow contributing to some of their other features. So how can you tell if you can help the anxiety to help them? If you hit it with something very strong, like benzodiazepines, they do calm down. So obviously everything, you know, will be subdued. But typical anxiolytics of the SSRIs rarely work. If they work, they may work for a short period and then the symptoms come back. So I don't think we've ever been able to treat the anxiety effectively to know how much of some of the additional phenotypes are due to anxiety.
There's another question. Thank you for that answer, by the way. There's another question. It made me, thank you for the wonderful talk. It made me think, can Rett animals procreate? What do you think would happen to offsprings of an ASO treated patient?
Great question. So Rett animals, the females, right, who have Rett, they're mosaic, they can procreate, and they're not great mothers, so we have to help them. And then part of the reason they're not great mothers, which really, you know, comes back to your question about anxiety, some of the things they do as the Rett females as mothers, they may eat their pup or hurt their pup, but they're anxious. And I think if we put with them another experienced mother, that helps a lot. So they can procreate. Even we know, sadly, of some human patients, not intentionally, who got pregnant and had children, and some had Rett child. So human females with Rett can have children as well, sadly. And what would happen to someone who's treated with an ASO? Let's take the best possible scenario. If you took a child with a duplication who is very sick, but you give them the ASO, they become functional, normal, live in society, right? If that person was to have a child, every female child of theirs, the boys will be okay because they'll get a Y chromosome, but every female child will be a carrier of that duplication. So in that case, that person may choose pre-implantation genetic diagnosis because ASO is not going to help the genetic material. It will only help the product, the RNA.
Thank you again for that. Other questions? Feel free to raise your hand if you want to just speak, or type your question in the chat box if you prefer that. There's another question. If I remember correctly, you showed in your summary that both the 70% and the 150% MECP2 mice showed anxiety behaviors, but linking that to your previous answer regarding chromatin structure, is the mechanism behind how this behavior appears with both chromatin contexts known?
Very good question. And let me maybe rephrase your question a little bit. I think you caught on something very interesting. Most of the behavior I showed go the opposite way, right? Gene expression goes the opposite way. Learning, Morotarod. The only one that goes the same way is the anxiety. And so your question is, what's driving that anxiety? If so many molecular things are going the opposite way, why is it that the anxiety going in the same direction? That's really a good question. I don't have an answer now. If I learn something, at least from the studies we're doing in the loss of function and as well with the ASO treatment, if I learn of a biomarker or something that will help us with that, that would be great. I'll share one tidbit with you that's really interesting. So I mentioned to you the anxiety couldn't be reversed if you were to treat in adulthood, right? We've let these animals be adult and be treated. One thing we noticed, if the animal did not grow with an anxiety, if the anxiety happened later on in life, it's reversible. How do I know that? It's a long-winded answer, but it's really interesting. It's cool. Since you have smart students, they have to listen. So one thing we wanted to know, if we took an ASO and gave it to a wild-type animal, healthy adult wild-type animal, let's give too much ASO, bring down MECP2 almost to nil, and see what happens. Of course, you're going to give them severe disorder, right? We want to see what's the worst case scenario. So you knock it out. Now the animals, as adult, they were born healthy, but as adult, they develop anxiety and all the features of Rett syndrome. Now the ASO with time will disappear. MECP2 levels will go back up. That anxiety was reversed, which is really interesting, which means it may be more a circuit issue, which is if an anxiety happened in childhood or early in development and postnatally, maybe the circuits change enough that it may be hard to reverse. But if you acquire it as an adult, it's more reversible. I didn't know that's hypothesis, but that's what the data showed. So it may not be chromatin, it may be really circuit.
Thank you. And there's another question. How different are the phenotypes from individual to individual? If they are very variable, how will you address it in a clinical trial?
Great question. And we're actually figuring out maybe why they're different. In general, they're very similar. So if you take people with duplications, they're going to fall in 2 to 3 buckets. They're all going to have motor problems. They're going to have seizures. They're going to have learning problems, language problems, and so on. But some may have worse seizures than others. Some, their motor problems will keep them from walking, while others will walk. Some will be bedridden always. So what we are learning is it has to do with the genomic locus, because some of them have actually duplications and triplications. So if you have a little bit more protein or a little bit less protein. So what we're doing now, to answer your question, we're going to classify people based on the level of the protein. And we're going to do protein level to phenotype relationships. And this way you'll know who to enroll in the clinical trial. The general features are all the same, but the degree of severity varies. And we think the protein level is the determinant of that severity.
Thank you again. There's a hand raised. Alana, please go ahead.
Hi, sorry, I have another question. I have many, but I know if I don't ask this one, I'm going to be wondering about this. You showed with your duplication that the mice performed better on Rotarod, and I know you didn't say it was like a motor discoordination, but I was thinking about that. I mean, generally when we see that, we think that this is an enhancement of performance. So is that, do you feel that they have an enhanced motor coordination? Is that?
So it's really hard for me. I try, all I can say is they're different from wild type. It's hard for me to interpret it. You know, Tom Sudoff calls it an autism-like phenotype of perseveration because he studied neuroligin mutants, and others have studied some autism models that showed they stay longer on the rotor rod. Certainly their coordination is fine. And why they stay longer, I don't know. Why do they do better on the contextual fear test? Why is their LTP much stronger? So everything seems to be that, but yet the final outcome is unfortunately neurological dysfunction in the patients. So it's something we haven't figured out, but we know that it can be reversed. The only thing that's really, and both of them have seizures. One thing I should mention to you, and I didn't go over these studies, we did look at the circuit. We began to look at the circuit. And the one thing we saw, although they're different, the two animals at every level, you know, if one has too little EPSCs, the other one has too much EPSC. Everything they're the opposite. But there's one thing they share in common, and that is they have increased probability of neurons firing together. And we call that increased synchrony. So when we did 2-photon imaging and looked at the circuit, we see the neurons tend to fire more together. And that may interfere with learning and may interfere with other features. But you're right about the rotor rod. So I don't know that it's improved function or just, I call it altered.
Thank you.
Question, is it expressed in glia and is it important in glia? It is to a much lower level. It's maybe 5 to 6 times more in neurons in glia than glia, but Gail Mendel has done very nice studies where she showed that if you rescue it in glia, you can improve breathing. So the breathing phenotype could be improved. Now, you know, on the other side, if you take it out, as you can see, we took it out in inhibitory neurons or in excitatory neurons, and either way you cause death. So obviously there, its presence in the glia is not sufficient to prevent them from dying. But it probably has some function in glia to improve, to help with the breathing phenotype.
Thank you. There's one other question there. Could the anxiety be related to lack of self-confidence? Perhaps the mice are afraid that they don't have the motor skills to not fall off the wings, for example.
Oh. So you see it in more than one task. You see it in the open field where they don't go into the center. You see it in the light dark where they don't go into the light side. We usually don't rely on one assay. So I mean, I don't try to think a little bit what the mouse is thinking about its confidence in a movement, but I think maybe given that we see it in more than one assay, it probably is at least more anxiety-like than some other, but we can't exclude what's driving it, right?
And one other question, is MECP2 affecting the expression of perineuronal nets around PV interneurons?
Very interesting question. A wonderful student in my lab who did an adult knockout study looked at that a little bit. Nothing was obvious, so we did not pursue it in greater depth, but I wouldn't claim that we've done it thoroughly. So one thing I should mention, I didn't have time to bring in the talk. You recall I hinted that maybe the delay in the onset of Rett syndrome in girls is due to the NCA mark being laid after postnatally. But now that we know we did an adult knockout, now that we know even if you come and knock it out in the adult, you take time before the phenotype appears. And even if you rescue, take time before it corrects. I think there's something else. I just want to make that clear. So while MCA contribute to pathogenesis, I don't think it explains for us exclusively, it explains the delay because we see the delay in adults, even after the MCA mark has been there and stable.
Thank you. There's probably time for one more question. Anybody has a burning question, please raise your hand or type your question in the chat box.
Given how great your question is, there's days for questions. You can always email me a question. I love these questions. Everybody is so wonderful. How far are we from clinical implementation? Oh. It really all depends if we get that biomarker. So everyone is working hard for that. It's important. We have the proof of concept. You need the safety where you have something to measure reliably to be sure you're not overcorrecting.
There's another one. Are you looking for a protein or mRNA biomarker, a blood or CSF biomarker?
Both. All of the above. We think CSF would be more reliable because it's more proximal to the brain. You want something as fast as possible and as proximal to the brain as possible. So we're thinking of, we're focused on CSF, and that's the clinical readiness trial. That's part of the study. They're enrolling patients collecting CSF.
All right, I think that will conclude our question and answer session. Thank you very much for taking the time to answer all the questions, and of course for the great lecture. Really brought everyone together in the middle of this pandemic and illuminated us with your work. So thank you very much, Professor Zogby. I want to also again thank Professor Graffstein for sponsoring this lecture series. This is my first time having the honor of introducing everybody and participating in it. So I'm very grateful. I think the massive attendance also speaks to the great interest in the lecture and the great appreciation in the lecture series. So I want to thank everybody for their participation as well and for the great questions, and wish you guys a great rest of the day and a healthy and happy weekend.
Thank you so much. And thank you so much to Dr. Graffstein and for the committee who selected me to give this lecture. It's really been a joy. I have to say, you know, I've given many lectures, but the level of questions and the breadth and depth of the questions and thoughtfulness is just, I could stay a whole hour talking with all of you. So thank you again for being so engaged and so brilliant.